Hi, my name is Sonalia Aluri.
I'm Karen Ruiz.
And I'm Leslie Cortez.
Today we're going to be talking to you about the electron transport chain.
The goal of the electron transport chain is to create ATP.
And ATP is the major energy source for the body.
The electron transport chain begins with complex 1, also known as NADH dehydrogenase. It is a large protein complex found on the inner membrane of the mitochondria. It accepts high-energy electrons from NADH molecules. The NADH molecules donates the 2 electrons onto an acceptor group called flavin mononucleotide, otherwise known as FMN, which is then reduced to FMNH2. Through oxidation-reduction reactions, the electrons move along a series of iron-sulfur groups. As the electrons move through the FeS clusters, the complex uses the electric current to pump 4 H+ ions into the inner membrane space. Coenzyme Q accepts the electrons and 2 hydrogens from the matrix to become the fully reduced co-QH2 form, and it then travels to complex 3. Complex 2 is a protein complex that accepts electrons from FAD and reduces it to FADH2. The FADH2 remains attached to the complex and gives off 2 electrons to a series of FeS clusters that ultimately transfer them to CoQ. CoQ accepts electrons and extracts 2 hydrogen ions from the matrix to become co-QH2. It then travels to complex 3. No protons are pumped, which is why less ATP is created from FADH2 than NADH. Next in the cycle is complex 3, otherwise known as cytochrome reductase. It catalyzes the transfer of electrons from ubiquinol to cytochrome C. It contains 3 important structures: cytochrome C1, cytochrome B, and Rieske center, which is a 2Fe2S group. The QH2 from complex 1 and 2 binds to complex 3, and the 2 electrons follow different paths. 1 electron moves to the 2Fe2S group of the Rieske center and is then transferred onto the heme group of cytochrome C1. It is picked up by cytochrome C, which is a water-soluble protein and can carry only 1 electron. It diffuses away and travels to complex 4. The second electron moves onto the heme groups of cytochrome B before being picked up by ubiquinone, which becomes partially reduced to form semiquinone. This pumps 2 hydrogen ions. A second QH2 transfers another pair of electrons which follow the different pathways as before, except now a ubiquinol is generated at the end. This pathway also pumps 2 more hydrogen plus ions for a total of 4 hydrogens pumped. The reduced cytochrome C then travels to complex 4. Complex 4, otherwise known as cytochrome C oxidase, accepts the electrons from cytochrome C and uses them to reduce oxygen and produce water. It transfers electrons from reduced cytochrome C molecules to oxygen. It contains 2 heme groups, heme A and heme A3, and 3 copper atoms. 2 reduced cytochrome C molecules give off a total of 2 electrons. 1 electron stops at the CuB group, which the other stops at heme A3. Once the heme A3 and the CuB are in their reduced form, an oxygen molecule can bind and abstract the 2 electrons to form a peroxide bridge. 2 more reduced cytochrome C molecules transfer an additional 2 electrons. 2 H+ ions are obtained from the matrix to help break the peroxide bridge to form CuBOH and heme A3OH. 2 more protons are extracted to oxidize the heme A3 and the CuB into their original state and releases 2 water molecules. In the process, 4 hydrogen protons are pumped. The pumping of hydrogens into the intermembrane space creates a proton gradient. ATP synthase allows the movement of hydrogen ions back into the mitochondrial matrix. As the hydrogen ions move down, ATP synthase phosphorylates ADP into ATP. A single NADH forms 3 ATPs, while a FADH2 forms only 2 ATPs, and that is the end of the electron transport chain.